Hello students, I welcome you to EPG Pathshala. My name is Dr. Shivkumar Sood. I am Senior Scientist in the Department of Animal Biochemistry at National Dairy Research Institute, that is ICAR NDRI in Karnal, Haryana, India. Today I am going to talk about protein structure hierarchy from the paper Biostatistics and Bioinformatics. An amino acid has 4 groups attached to a central carbon in tetrahedral conformation known as C-alpha. The central C-alpha atom has 3 common groups attached in each of the amino acids. The amino group is bonded in direction going below the plane of the paper, shown with dashed wedge in this slide. Second group is a carboxyl group bonded in direction going above the plane of the paper, shown with solid wedge in this slide. Third is a hydrogen atom shown with normal line to convey that it is bonded in the plane of the paper. The fourth is a variable side chain in each of the 20 naturally occurring amino acids and is shown with the normal line to convey that it is also bonded in the plane of the paper. There are 20 naturally occurring amino acids. At the root level, the amino acids can be classified in 3 groups. First is unique group having amino acids glycine and proline. Glycine has attached only hydrogen as the side chain, and proline is attached with 3 methylene groups chain attached to both C-alpha and nitrogen of the amino group to form an amino acid starting with I as opposed to A, amino acid versus amino acid. The second group has polar side chains either uncharged at neutral pH or side chains having charged acidic or basic groups. The third group has non-polar side chains either aromatic or hydrophobic. The second group with polar side chains has sulfur containing amino acids methionine and cysteine. The second group with uncharged hydroxyl group at neutral pH include serine, threonine and tyrosine. The second group also includes amide group containing amino acids asparagine and glutamine. The second group with polar side chains having charged acidic group include aspartate and glutamate. The second group with polar side chains having basic groups include histidine, lysine and arginine. The third group having non-polar aromatic side chains are present in phenylalanine and tryptophan. The third group having non-polar hydrophobic side chains may be slightly bulky as in leucine or isoleucine, or small side chains as in alanine and valine. In the helix shown here, only main chain atoms are shown. In this structure, each peptide nitrogen is shown as blue, C-alpha as black and peptide carbonyl oxygen as red. In alpha helix, every current amino acid is hydrogen bonded to 4 residues ahead, that is every 5th residue in the chain. This helix is classical alpha helix. Dear students, in this module, we have seen the hierarchical folding of the proteins having 4 levels. At the first level, we have seen the formation of the peptide bond through covalent amide bonds. At the second, that is secondary level, we have seen the folding of the main chain into alpha helices and beta strands, which are extended strands. Then these 2 secondary structure levels, they are joined together through what we have named as loops. The secondary structures which are regular are alpha helices and beta sheets, or beta strands, and which are not repeating phi and psi having values, they are loops. Then these secondary structures, they are tightly packed within a core into 3 dimensions, so that it has a surface which is labile, and this is the surface which is having the functional sites like active site of an enzyme or binding site for a receptor. Sometimes you see the secondary structures may have defined pattern, for example a helix, then a beta turn helix. This is called supersecondary structure. Then the tertiary structure subunits or the independently folding domains, they interact with each other through non-covalent interactions and form quaternary structures, and through this quaternary structure the main regulation is achieved, that is the allosteric regulation. So the enzymes which are having quaternary structure, they have evolved for the regulation of their function. For example, hemoglobin, which is having 4 subunits, is a allosteric protein. I thank you all for visiting EPG Pathshala. Thank you.